This is a very quick review of where fatty acid or lipid metabolism, in your diet, okay, whatever you're eating, if it contains lipid comes in, okay, goes through your stomach and then it enters your intestines, okay? And in your small intestine, you secrete these bile acids. Remember, we talked about bile acids being molecules that can sort of solubilize, in a way, the triacylglycerides. What happens is that after they are sort of digested and your lipids are going to pass through intestine and enter the bloodstream. And they exist in these forms called the chylomicrons, all right? And what these things are a mixture of triacylglycerides plus lipoproteins, okay? So they have a bunch of proteins and a bunch of lipids inside them. And then they get to be transported through your capillaries, and then capillaries, and then they get passed on to other parts of your body, okay? Like your peripheral tissues, whichever places that could use some lipids, it gets passed through there. And after the usage, you do get remnants of chylomicrons and cholesterol, and usually what happens is that they enter the liver, okay? And then the liver can sort of reassemble them, group them into larger lipoproteins, and then they can come out, utilize again, okay? And then the leftovers can then again go to liver and then comes out, okay? So the process repeats, okay? Now, remember we said that in this entire process, you will get some low density lipoproteins, and those are what we call bad cholesterol for you, okay? So if you go to the hospital and you analyze your blood, okay, and there are several things that you look for, like triacylglycerides, lipoproteins, okay? If you get a blood test, and if your low density lipoproteins level too high, the doctor is going to tell you, yeah, you need to do exercise and then, get rid of the low density lipoproteins. Because usually it's these things that will accumulate inside of your arteries and cause blood circulation to cease, okay? All right, so today let's begin with the mobilization of stored triacylglycerides, right? So we talked about this is how you digest them and how to sort of transport them and disperse them into your body, okay? Now, we talked about how lipids are typically stored in adipose tissues or adipocytes, okay? Adipocytes, and typically they're stored there until you need to use them, okay? Now, when we need to use them, how do we get them out, okay? So when you are low on sugar or when you're low on energy, your cell is going to secrete hormone, which then will bind to your adipocytes, okay? And through a reaction pretty similar to what we talked about in the signal transduction with glucagon and insulin, that kind of stuff, right? So what you have is a receptor on the cell surface, and if it attaches to your hormone, it's going to induce a conformational change of this enzyme, okay? And then it's going to turn on some G proteins, okay? So you probably heard about G proteins in general bio. And then this G protein can then activate the adenylyl cyclase, okay? Which then can turn ATP into cyclic AMP, okay? And we talked about how cyclic AMP is sort of like an intracellular regulator that is used a lot to trigger different responses, right? And guess what? Cyclic AMP, okay, will activate, okay, so this is activate. It's going to activate this thing called the protein kinase A, okay? So it's going to activate protein kinase A, right? And then protein kinase A can do two things which will allow you to utilize the stored TAG, okay? Number one is it's going to phosphorylate, okay? It's going to phosphorylate these proteins called the perilipin. And perilipins are these proteins which sort of surround your oil droplets, right? So if your protein kinase A, because it's protein kinase, right? Remember we said blah blah kinase. It's an enzyme that can use ATP to phosphorylate the blah blah. Okay, so protein kinase in this case can put a phosphate from ATP onto this thing, and then it'll become this, right? And once it's phosphorylated, of course, it also is going to undergo a conformational change, so the shape changes a little bit. And as you can see from the cartoon here, that initially it was all horizontal and closely packed. If it's phosphorylated, you get these sort of holes between the perilipin, okay? And then that's where your lipase can go in and react, okay? Now, the protein kinase A also does another thing, which is to activate your lipase, right? It's going to turn on this hormone-sensitive lipase. Again, it's just a lipase, okay? All I care about is you understanding what a lipase is. It's called the hormone-sensitive lipase because it's turned on by the binding of a hormone from the outside, okay? So that's just how it is, all right? So your protein kinase A will do two things. One is it's going to phosphorylate perilipin, which will then make your TAG accessible. That's number one. And number two, it's going to activate, okay, or turn on your lipase, okay, by phosphorylating it, and then the lipase becomes active, okay? And once the lipase, this guy, okay, if this guy is active, then it's going to get close to the droplet, the lipid droplet, and it's going to extend a little arm into here and start doing the hydrolysis, okay? So then it can break away your TAG, liberating fatty acid and glycerol, okay? Because that's what a lipase does. It converts TAG into glycerol and fatty acid, okay? Now, the liberated fatty acid can then exit your cell and go into the bloodstream, which then you have serum albumins. If you guys, I don't know, have you guys done BSA quantification in biochem lab yet? Or will you guys even do that? Okay. BSA, it's a bovine serum albumin. It's a protein that we use in the lab a lot. You put that in your cell culture medium sometimes, okay? So it's just a protein. It's a protein inside of your bloodstream, right? So these albumin type of proteins, and they can attract these fatty acids, and these fatty acids get, go along the bloodstream, and it's going to get transported until where it's going to find the cell that it wants to go into. For example, myocytes, right? So muscle tissues, and you have these fatty acid transporters, which then will take in the fatty acid into the cell, and then you can then utilize this fatty acid, okay, through what we're going to talk about next, this process called the beta oxidation, okay? So you can oxidize your fatty acid, let it become CO2 and ATP, okay? So this is how you utilize stored TAG, okay? So first thing first, we know that TAG, when it's degraded by lipase, it's going to form glycerol plus 3 fatty acid, okay, using water. All right, so glycerol metabolism, we talked about that when we talked about sugars, right? Glycerol looks like that. It's 1, 2, 3 carbons with OH on all 3 carbons. So if you want to metabolize glycerol, the easiest thing would be to convert that into your sugar triose, okay, your triose. And the closest would be DHAP, which all you have to do is oxidize C2, right? Because here initially it's alcohol, and then here it's a ketone. So you did an oxidation, and then you would phosphorylate C3 Okay? Why? Because here initially it's a alcohol. Here now it's a phosphate. Okay? It's a phosphoryl group. So how do you get the phosphate on? You have to use ATP to do that, right? So it's 2 steps. One is going to be glycerol kinase. So this is a kinase. And then the other step has to do with oxidation. So this guy is glycerol 3-phosphate dehydrogenase. I'm only writing it because you can see it larger. Okay? All right, so glycerol 3-phosphate dehydrogenase. All right, so 2 steps. Kinase, dehydrogenase, and then you get your DHAP. Once you have DHAP, you have G3P. Yeah? Glycolysis, right? So fatty acid degradation, then it's going to be something new, right? So glycerol we talked about. Now, fatty acid, in order to degrade fatty acid, you have to first activate it, okay? Remember what I always say about organic acids? That organic acid, oh, by the way, when I say organic acid, typically it just means carboxylic acid, right? Because you don't really have other types of acids in biology. So typically when people say organic acids, a lot of chances they're going to be talking about carboxylic acids, all right? So again, remember that organic acids, for example, this guy, even though we draw it this way, but please do remember that in reality you have resonance. It is virtually impossible to differentiate the 2 oxygen atoms. As a result, we usually say it's like that, right? So usually when we draw organic acids, it's like that. Okay, now, because it has resonance, organic acids are like super stable. Okay, they're very stable. So if you want to do any chemistry on it, you better have some kind of activation first. Okay, so if you want to do chemistry on organic acids, you have to first activate it. Okay? Now, by activation, organic acids, okay, acids that are activated in biology, you only have 2 things. Okay? You can have using ATP to make it into an acyl phosphate, like a phosphoester. That's going to look like this. Okay? You can have a phosphate. This is a phosphor, and then this is ester, right? This guy is an ester, right? You can have a phosphoester, or, okay, it has to be thiol ester, okay? So in biology, you pretty much probably only have 2 or 3 choices if you want to activate an organic acid, okay? All right? You have to make it into some kind of ester in order for it to become activated, in order for it to be utilized. Okay? All right. Now, turns out in fatty acid activation, it's going to be like acetate activation. Okay, so all straight chain organic acids, if they're activated, what you'll get is you're going to use this thing called the fatty acyl-CoA synthetase. So again, it's a synthetase, not synthase. Okay? Remember, synthase does not utilize ATP. Synthetase uses ATP to, do some stuff. Okay? Now, fatty acyl-CoA synthetase, meaning it's an enzyme that makes fatty acyl-CoA using ATP. Okay, so great. Now, what you're going to do, this little thingy right here, this is the fatty acid. And on top, you have ATP. Okay? What the enzyme will do is have your fatty acid attack the first phosphate, okay? The first phosphate of ATP, right? And then you generate 2 products. One is called pyrophosphate. Usually we write this as PPI, okay? 2 phosphates inorganic. And you will also get this thing called the fatty acyl adenylate. Okay, so it's adenosine plus a phosphate group, right? So this thing, okay? Now, let's first talk about what happens to your fatty acyl adenylate, okay? Because this guy is like, this thing is an ester. What you can do is do a trans esterification. where you can use thiol to attack here. Electron go on top. When the electron comes back down, this thing leaves. Okay? So like the mechanism that we've been talking about so much. Okay? Now, when it does that, you do your transesterification kind of thing, and then what you are left with is a thiol ester. Okay? This is sulfur. Right? Now, what you want to remember is that you generated this PPI. Okay? And pyrophosphates inside of your cell, usually they don't stay as pyrophosphates. They will become 2-phosphates by using this inorganic pyrophosphatase. Okay? And then it splits into 2 organic phosphates. Okay? And if you look at the energetics of this entire reaction, the pyrophosphate becoming 2 inorganic phosphates, you have about minus 20 kilojoules per mole. And this entire activation, the ATP-dependent activation, gives you about minus 15 for both 1 and 2. Okay? So then, together, your net delta G is about minus 34. Okay? In this process, you actually consumed 2 ATP equivalents. Okay? Does that make sense? Why? 好来，所以 fatty acid activation， 你看这个图应该可以接受嘛，对不对？就是你今天有一个 fatty acid， 那 fatty acid 呢它是有机酸嘛，那有机酸呢它非常的稳定 ，OK， 所以如果你要做后续的 chemistry 在有机酸身上的话，你需要先将它活化 ，OK， 那活化的机制通常在 biology 里面大概就是2种，一种呢就是拿这种 phosphate 类似的东西插在它身上，那它就会变成这种 phosphate 的 ester，OK， 那另外一种做法，更常见的做法就是你把口 A 放在它身上，那你就会变成一个 thiol ester。那一旦它是有一个 ester 的情况下，它的能量会比较高，所以它可以去做一些它要做的事情。OK， 那这边呢要讲的是说，你fatty acid 做 activation 的时候呢，它会 generate 出来的是这个一般我们在讲 kinase 啊，你通常吐出来是1个 phosphate。OK， 通常是1个 phosphate。那这边呢，你吐出来不是1个 phosphate， 你是吐2个 phosphate 出来。OK， 所以这边的 net reaction 呢 ，OK 是画个小方块在这边。OK， 所以你这边的 activation 呢，一般的我们说 kinase， 它是拿 A 加 ATP， 它会变成 A 上面带一个 phosphate， 然后加上 ADP， 对不对 ？OK， 或者是说。你今天做一些需要用 ATP 的 enzyme， 你也会是比如说 A 加 ATP， 然后变成 B 加上 ADP 加 PI。OK， 这是一般我们说用 ATP 的 enzyme 会做的事情。OK， 但是在这个 case 呢，你做出来的是原本是 fatty acid 加上 ATP， 它会变成 AMP 加上2个 PI， 再加上一个 fatty acyl 的口 A，OK， 那注意这边是 AMP 加2个 PI，OK， 那各位要记得 ATP 到 AMP 啊是 the same thing as 2个 ATP 到2个 ADP， 主要的原因呢是因为你看哦，我们说 AMP。你要回去 ATP 的话，你必须要 AMP 先吃一个 ATP， 然后 ATP 会变 ADP， 然后 AMP 会变 ADP。OK， 所以 anytime 你只要有一个 enzyme， 它是 AMP forming， 就说它吃完 ATP 之后吐出来的变成 AMP 的时候，它对我们来说，它等于是吃掉了2个 ATP。Right， 因为 ATP synthesis 有没有？我们记不记得我们上个1/3在讲 ATP synthase 的时候，它是 ADP 变 ATP 嘛，不是 AMP 变 ATP。OK， 所以如果我们要去算 ATP equivalent， 就是说你这个 process 消耗掉几个 ATP 的话，你必须要用 ADP 来算。OK， 懂我意思吗？可以了哈。Right， so fatty acid activation essentially causes you 2 ATP instead of 1. Okay? Couple of things. Fatty acids, how it's metabolized inside the cell. Okay, fatty acids being a large molecule, so it has to have some kind of transporter. Okay, now turns out if the fatty acid is carbon length 12 or lower, it can be transported directly into mitochondria through a transporter. If it's longer, like 12 and above or 14 and above, you have to use this thing called the carnitine shuttle. Okay? And carnitine shuttle is just a mechanism which you can bring in the fatty acid by tagging it to carnitine. Carnitine looks like this. Okay? This is carnitine. And essentially what happens is that in your cytosol, okay, your fatty acids here, okay, it's going to first get activated into fatty acyl-CoA. Okay, so this is the activation. And fatty acyl-CoA can then react with carnitine and liberate the CoA, right? And you form this fatty acyl-carnitine. So all you did was you changed the carrier from CoA to this thing called carnitine, and in this entire process, you turn the fatty acid from cytosol into the intermembrane space. Okay? And then what you're going to have is carnitine, your fatty acyl-carnitine then can then interact with your CPT2 and do the exchange again with the CoA inside the matrix, and then it becomes, okay, and then it becomes fatty acyl-CoA inside of the matrix. So this guy is mitochondria. Okay, and then this is the intermembrane space between mitochondria and cytosol. Okay, and then now this guy can then be used for degradation. Okay, does that make sense? So all you have is just a little transporter in the middle that can, take the fatty acyl portion of the molecule and then translate that into mitochondria. Okay, so just a little transporter. Don't need to worry too much about that. All right, now, the overall pathway of beta oxidation, I'm going to show you the reactions one by one. Now, this is sort of the summary. Let's start with the summary. For every round of beta oxidation, you will generate one acetyl-CoA, one NADH, and one FADH2. The way it works is like this. Okay, let's say you start with a C16 acyl-CoA. You end up with a C14 acyl-CoA plus acetyl-CoA. Okay, so every cycle, okay, or you can say every time you run beta oxidation, you cut out 2 carbon. Okay, so initially you start with 16, you end up with C14 in one cycle, like 14 plus 2. Okay, all right, now the reason why it's called the beta oxidation is because all of the chemistry is happening on the beta position. Okay, first step, you do a dehydrogenation. Okay, and what you're going to do is you're going to turn initially here between C2 and C3. Initially is carbon-carbon single bond. Now through the dehydrogenation, if you look at here, now it's carbon-carbon double bond. So you did an oxidation. Okay, and in this process, the reducing cofactor that you're using is FAD. Okay, FAD becomes FADH2. And remember I said, I think at the very beginning of our class, I said this thing where, NADH is soluble, so NADH is found floating around in the solution. But what about FADH2? Okay, FH2 usually you do not see it as a free substrate. Okay, FADH2 is usually a cofactor or a prosthetic group kind of inside of an enzyme. Okay, so FADH2 is usually always inside of an enzyme, whereas NADH can float around in the solution. 听懂我意思吗？这很重要哦。Okay. Now, because FADH2 is usually inside the enzyme, so it has to interact with some other enzyme in order to pass the electrons down. Okay, so in this case, what turns out happening is your FADH2 here is going to directly interact with, what do you think these things are? What is this? Q becoming QH2, electron transferring chain, right? So this guy is the ETC. Okay, so it directly interacts with the ETC. So if you go back to the lecture where we talked about the ETC, right? Remember I had one slide that says, you know, NADH can interact with the ETC, FADH2 can interact with the ETC. You know, what else can make Q becoming QH2, right? And remember there was one thing where it says like fatty acid degradation become FAD through this enzyme that carries the electron, and that enzyme interacts with the membrane, right? So this is what I was talking about back then. So what happens is that this enzyme, okay, this enzyme, which I don't have the name for you right now, but this is some kind of dehydrogenase. Inside of this dehydrogenase, you have this FADH2, and then FADH2 will pass the electrons to this thing called the ETF, the full name, which I will talk about a little bit later, but the full name for this thing is electron transferring, what's the F? Flavoprotein. Okay, it's a protein that contains flavin molecules like FAD. Okay, so it's the flavoprotein. All right, doesn't have anything to do with flavors. Okay, just so you know. 不是说什么吃起来会变甜甜的。All right. And this electron transferring flavoprotein can then interact with the ETC, okay, and dump the electrons from FADH2 onto Q, becomes QH2, okay? Now, once you have an alkene, it's a lot easier to do chemistry with, okay? Alkene can do what? Okay, you can hydrate at the beta position, alpha, beta, okay? And you do a hydration, okay? You use water, you hydrate it. Now you have beta position OH. If you have OH, what can you do? You can dehydrate, which becomes the alkene. What else can you do? You can oxidize that. Okay, you can oxidize OH. So you're going to do another dehydrogenation. And now you oxidize this guy. So you turn it from an alcohol to a ketone. Okay? You oxidized it. And then the next step is kind of cool. Okay? The next step, now you have this diketo kind of thingy. Okay? You have two ketones and then you have a carbon in the middle. Okay? This thing is called the beta keto, or sometimes people call it 3-keto. Why? Because guess what? The CoA here is 1. This guy's 2. And then this carbon is 3. Okay? So 2 is always the same as alpha. 3 is always the same as beta. Okay? Make sense? All right. And then once you have the beta keto acyl-CoA, and what you can then do is do a thiolytic cleavage using CoA here. And the end result is you chopped it in half. And you liberate acetyl-CoA plus 14 carbon acyl-CoA. Okay? Obviously, the 14 carbons can, this guy, can like, whoa, come back here and do the entire process again. After the second time, you have 12 plus 2. The third time, you have 10 plus 2. And then another time is 8 plus 2, 6 plus 2, 4 plus 2. Okay? And then at the end of 4, you only have 2 plus 2. And the 2 plus 2 are 2 acetyl-CoA. 可以吗？可以了哈。Okay? Alright, so let's talk about the reactions in detail. Alright, the first reaction is catalyzed by this thing called the acyl-CoA dehydrogenase. I mean, come on. The substrate is acyl-CoA. The enzyme is a dehydrogenase. So the name is acyl-CoA dehydrogenase. Okay? And it's going to catalyze the removal of 2 hydrogen atoms. Okay? So 2 hydrogen atoms is essentially your H2 Okay? From the alpha and beta carbon, which then will generate a trans alpha beta unsaturated acyl-CoA. Okay? So the product, we usually call it trans enoyl-CoA. Okay? Because, you know, it has an alkene inside. In most animals, okay, turns out you have 3 different isozymes. Isozymes mean that they are pretty much the same enzyme, but they're encoded by different genes. Make sense? Okay? 3 isozymes, and the 3 isozymes differ in terms of their preference for chain length. Okay? Remember last semester we talked about substrate specificity? Okay? Like chymotrypsin for phenylalanine versus glycine, right? Size matters. 听起来有点奇怪, but size matters, okay? What happens is that you have a very long chain, VLCAD, very long chain acyl-CoA dehydrogenase, or you have a medium chain acyl-CoA dehydrogenase, or you have a short chain acyl-CoA dehydrogenase, okay? But they're same enzyme, okay, same type of enzyme, but they have different substrate preference, okay? Some like to react with the longer substrate, some like to react with the shorter substrate, okay? That's just how it is, all right? And the reaction goes like this. The pro-R hydrogen on the beta carbon, okay, is transferred as a hydride equivalent to FAD, okay? So it's always the pro-R here. Why? Just because the enzyme is 3D, so enzyme, it just so happens that the active site likes the pro-R. So that's just how it is, okay? So what you can do is you can first deprotonate your alpha carbon's hydrogen, okay? You can deprotonate that. What's the reason why you can deprotonate on the alpha carbon? Resonance, okay? Alpha carbon 要记得 alpha carbon 可以被 deprotonate， 好不好？就是我很难再解释为什么了，因为它会有 resonance， 因为你 deprotonate 它之后呢，这个东西这个 electron 可以 share 到这边，这个 electron 可以 share 到上面，所以你可以出现一个这个 enolate， 有没有 ？OK， 那因为这个是 resonance 嘛 ，OK， 这就是为什么 alpha carbon 身上的 proton 可以被拔掉，其他 carbon 身上的 proton 没事，不要去拔它 ，OK？ 应该说你也拔不掉 ，right? But just you know, I'm just saying this, right? Okay, so once you deprotonate your alpha proton, then you have this thiol ester enolate. Okay, this is a minus charge here, and what the minus charge can then do is it's going to form a carbon-carbon double bond. Now this carbon, your C3 carbon here, has too many bonds, so as a result, it's going to leave onto FAD as H with 2 dots, okay? So it's going to leave as a, this guy is called hydride, okay? Hydride, H minus, okay? And the product is trans-enoyl-CoA, okay? So you went from saturated acyl-CoA to unsaturated acyl-CoA, right? So this is how it works in this enzyme. This acyl-CoA dehydrogenase enzyme, inside it you have FAD. So you're gonna pass the electron to FAD, and obviously this FAD becomes FADH2, okay? And then you have another enzyme, this guy, ETF, stands for electron transferring flavoprotein because it has what? Flavin. Okay? And basically FAD. Okay? It's a flavoprotein. Alright, so you're going to take the FADH2, pass the electrons onto this FAD. So this FAD becomes FADH2. Okay? And then this protein can then interact with the ETC. So this whole thing is the ETC. Okay, and it's going to pass the electrons onto CoQ. Okay, and then it's going to go through your electron transferring chain. Eventually, the electron reaches molecular oxygen and generates water. Okay, great. We're good. What you notice is something similar. Is that if you look at this part, the fatty acid degradation, and if you look at succinate, this guy is succinate to fumarate. Remember, it also generates FADH2, right? So this reaction is very, very similar to the succinate dehydrogenase, right? Because remember, succinate to fumarate, what's fumarate? Fumarate 是什么？有没有记得 fumarate 长什么样子？不记得。那你觉得 succinate 的下一步会是什么？加 OH 吗？没那么快，加 OH 前面还有一步。哎，对。Okay, alkene. Okay, so what does this look like? 这个是不是就跟我刚刚讲 beta oxidation 的第一步基本上是一模一样的反应，有吗？所以这个 reaction 其实是一模一样的反应，只是它吃的 substrate 不同而已。所以它们的 enzyme 也就会非常非常的类似。Okay, all right. Reaction 2 and 3, you have eno-EO-CoA hydratase. So it's a hydratase. Hydratase meaning I add OH onto something. So it's going to add OH to the alkene. It's going to hydrate the alkene. And then you have this 3-hydroxy-acyl-CoA dehydrogenase. It's a dehydrogenase acting on 3-acyl-CoA. Okay? So turns out it's absolutely identical to the TCA cycle after the oxidation of carbon-carbon single bond to carbon-carbon double bond. The next step is hydration of the CC double bond to 3-hydroxyl. And then the hydroxyl is then oxidized to a keto, just like TCA cycle. Okay? So TCA cycle, you have succinate to fumarate. In fatty acid degradation, you have a saturated fatty acid to the alkene, eno-EO-CoA. Okay, so alkene. Next step, you have hydration. In TCA, you go from fumarate to malate. In fatty acid degradation, you have eno-EO-CoA to 3-hydroxy-acyl-CoA or beta-hydroxy-acyl-CoA. Same thing, okay? 你要叫它 beta 或者叫它 3 都可以, okay? And then you have oxidation. 在 TCA cycle 里面，你是 malate oxidized to oxaloacetate OAA, okay? At the OH position, and then in fatty acid degradation, you go from 3-hydroxy-acyl-CoA, oxidize that using 3-hydroxy-acyl-CoA dehydrogenase. It becomes 3-keto-acyl-CoA. Okay, so almost identical to the TCA cycle. Okay, so if you look at it carefully, all it is how do you go from a carbon-carbon single bond from alpha-beta carbon next to a carbonyl into a oxidized C3, okay, oxidized carbon 3 to a ketone. Okay, so let me draw out more carefully. Okay, so again, this entire process, you go from, we say 1, 2, 3, or you can call it alpha, 3 is beta, okay? And the first step is always, you want to, because we're in the degradation mode, okay? Degradation means oxidize something. Remember that? Degradation is equal to oxidation. Okay, so first thing you're going to oxidize. Like that. Okay, you have to first generate the alkene. Without the alkene, you cannot do OH. Okay, we will talk about how to do that, but just for most biological purposes, in this case, you have a carbonyl here. Because you have a carbonyl here, you can do the alpha carbon deprotonation. Because you can do alpha carbon deprotonation, then you can very easily generate this dehydrogenation event. Okay? And then the next thing is to convert this guy through hydration. And at the 3 position, you have OH. Okay? And then you have another oxidation. Right, because initially this is alcohol, and remember there's a hydrogen here, there's no hydrogen here, right? So no hydrogen here, okay? Does that make sense? So you're doing here, this guy is called a dehydrogenase because it's going to dehydrogenate this hydrogen. Okay, that's why it's called dehydrogenase. Okay, and then you generate a ketone, and now when you get down to here, what you can then do is the next step, which we'll talk about, which is dialysis. Okay, you can then cut out this part. Okay, so the degradation pathways have a lot of things in common: TCA cycle and fatty acid degradation. Very similar. Okay, so repeatedly, what I hope you guys can sort of get a feeling for is in metabolism, it's pretty much the same reaction happening over and over, just different substrate. Okay, 所以真的生化会比有机好学很多，因为你会发现到说反应的 type 其实没有很多，反应的 type 其实很少，只是说他们吃的 substrate 不同而已。Okay. All right. Reaction 4 requires a little more detailing and attention because this reaction is the part where you're splitting carbon-carbon bonds. Right? So those of you that took my class last semester, this was your final exam question. Okay, what's the mechanism for thiolase? Right? So the fourth and the last reaction in each cycle of the beta oxidation involves the attack of a nucleophilic thiol. Okay, onto the electron-poor keto carbon of 3-keto-acyl-CoA, and it results in the cleavage of the bond and release of acetyl-CoA. Okay, so the other product of this enzyme is the shortened fatty acyl-CoA, and that's ready to begin a new cycle of oxidation. Okay, so the way it works is like this. This guy has electrons, right? And it's going to, very simply, it's going to attack here. Well, first of all, if this one is a nucleophile, where can it attack? It can attack here or here, right? Because those are the carbonyls. Now, some of you might have a question of like, wait, how come it doesn't attack here? Okay? Let's say if it attacks here, then you pop up the electron up here. When the electron pop down, you are only able to kick out this guy. Okay, and when this guy leaves, what is it? A CoA. Okay. So same thing. Okay. So it doesn't happen that way. Okay. So this reaction, no. Okay. No good. All right. So it turns out we're going to attack this carbonyl. Okay. So this is the enzyme reaction mechanism. Lone pair electron on the sulfur. Okay, so this guy is sort of the nucleophile. And because it has lone pairs, it's a nucleophile, it has a lot of electrons, so it can attack a electrophile, which is the carbonyl carbon. Okay? If it attacks it, you form a new bond. Now this carbon has too many bonds, so one of the bonds has to go somewhere. It goes up to the oxygen. Right? And you form this tetrahedral. Oxy and ion intermediate. Right? And then when the electrons pop back down, Now you have two choices. You can either pop away the CoA or enzyme. Actually, this is not the CoA yet. This is just the enzyme. Okay, so note here, this is enzyme. Okay, or you can pop the electrons onto this carbon. Okay, so turns out it does that when the electron pops it over to here, and what this will do is it's going to, essentially grab a proton, and then it becomes a methyl carbon. 可以吗? So far so good. Okay, so the electrons, when this electron comes back down, somebody has to go. Like this guy has to go, this guy has to go, or this guy has to go. Okay, 那这3个棒其中有一个棒要飞出去，那要飞谁？如果你飞的是 thiol 的这个 bond 呢，那你就回去到这边了，回去到上面，所以走的不会是它，所以走的呢 turns out 会是上面这个 bond。为什么？因为这个东西啊 ，check this out， right？ Electron here, 2 electrons, goes onto this carbon, right? What's the intermediate that you generate? 它的这一步出来的 reaction product 是什么？然后呢，上面是不是带一个 minus charge？ OK， 那这个东西为什么可以发生？就是为什么可以有这个 intermediate？ 这个会解释为什么它弹 electron 的时候不会弹到那个 R group 上呢？不会 spin 到，不会 throw the electron 在 R 身上，它一定会到这边。为什么 ？What is up with this guy? Resonance, okay? Where's the resonance coming from? This guy is alpha carbon 有没有？然后这个东西是什么？这个东西就是那个 alpha carbon 如果被 deprotonate 出来，不就是这个东西，对不对？如果我说，你看这个 structure， 这边是 alpha carbon 嘛，对，下面这边是 alpha carbon， 所以如果下面这个 carbon 呢，它被拔了一个 proton， 它是不是就变成上面这个东西？是嘛，对不对？那上面这个东西呢，它可以有 resonance， 这个 electron 可以跑到这边，它可以跑上来。那因为它有 resonance， 所以它会被 stabilized。所以就是你这边有1个 bond、2个 bond 跟3个 bond， 所以 electron 要去哪里？要去哪一个 atom 身上？当然你可以去 sulfur 身上，但去了 sulfur 身上呢，就回到你的起始物了。Okay， 所以你不会这样做。那 R 你没办法稳定它，所以它会 unstable， 所以不会发生。那 turns out 唯一可以选择的就是这一坨 ，OK？ And then as a result, what you get is acetyl-CoA, okay? Because once you have this thing, and this guy can then grab a proton, okay? And then it becomes acetyl-CoA. So acetyl-CoA is out. However, what you notice is that you still have a big portion right here, okay? Right here, that is attached to enzyme. Obviously, you have to get that one out. Otherwise, the enzyme is like trashed. Okay, so you have to get this other thing out. So to do that, you will do a trans-esterification using CoA. Okay, so you will have another nucleophilic attack onto the carbonyl carbon, and then you generate again tetrahedral oxyanion intermediate. Okay, and then when this minus charge comes back down, now you can kick out, of course, you can still kick out CoA, but then it will just generate this. So you don't want to do that. And turns out you will kick out the enzyme. Okay? So you kick out the enzyme. Enzyme is regenerated. And then this guy is your CN minus 2. Okay? Initially you start with CN. N could be like 16. The product is Cn minus 2. So like C16 minus 2 is like what? 14, right? So every time you do beta oxidation, you spit out acetyl-CoA. Okay? And this enzyme again is called the 3-keto thiolase. It's called thiolase because it's using a thiol to split the molecule in half. Okay? This is the overview of fatty acid degradation. Right? You start with fatty acid entering the cell. Now it's going to be in the cytosol. Okay? It has to get into the cell first. And then inside the cytosol, it has activation. Okay? So you will use ATP to activate that into fatty acyl-CoA. And again, check this out. you do make AMP instead of ADP. Because of that, this is equivalent to saying 2 ATP. And then fatty acyl-CoA can then go through this carnitine shuttle, entering, this is mitochondria, okay, where your degradation occurs in mitochondria, okay. Now in the mitochondria, it's going to regenerate your fatty acyl-CoA from fatty acyl-carnitine, and then it's going to do the beta oxidation 4 reactions, okay, 1, 2, 3, 4, dehydrogenation, hydration, dehydrogenation, and then kicking out acetyl-CoA, okay, thiolysis. Okay, this guy is called thiolytic cleavage. Okay, and then you generate fatty acyl-CoA, N-2 CoA. Okay, and then you do this again. Okay, so in terms of the energy yield, all right, so this guy is kind of important, right? All right, so activation, let's say you start with a C16 fatty acid, okay? Because why would we say C16? Because C16 fatty acid is the most common fatty acids in the diet, okay? C16 fatty acid, you want to degrade that into ATP, okay? What's the overall yield in terms of ATP? First, the activation of acid to CoA, you -2, okay? Because you spent 2 ATP. Now, you have C16, so this guy is C16 CoA. How many cycles of beta oxidation do you use to turn C16 fatty acid CoA into acetyl-CoA? 7次 ，OK？ 你跑7次哦，你会说，哎，为什么不是跑8次？16除以2不是等于 like 8吗？因为记得最后一次的时候，你吐出来就是2个 C2 了 ，OK？ 所以你就不会再跑第8次 ，OK？ So turns out you only run the fatty acid degradation 7 times. So if it does it 7 times, you generate 7 NADH, 7 FADH2, okay, and then 7 acetyl-CoA plus 1 acetyl-CoA left over. So total is 8 acetyl-CoA. Alright? Now, in terms of ATP, TCA cycle burns 1 acetyl-CoA into 2 CO2 completely. In 1 round of TCA cycle, you generate 10 ATP. Why? Because TCA cycle generates how many NADH? 来，我们一步一步算好不好？一步一步算嘛。OK，acetyl-CoA 跟 OA 加在一起变 citrate， 会不会产任何东西？不会，对 citrate 到 isocitrate， 不会嘛？对 isocitrate 到那个 alpha kg，alpha ketoglutarate， 然后会吐一个 CO2 出来嘛？记不记得 ？isocitrate 到 alpha kg 会产一个什么鬼 ？ATP、FADH2 还是 NADH？NADH 对，所以已经一个 NADH 哦。alpha kg 到下一步是什么 ？succoA 对不对 ？succinyl-CoA。alpha kg 到 succinyl-CoA 会产一个 NADH 对，2个 NADH 哦。然后 succinyl-CoA 到下面一个是 succinate 对不对？所以 CoA 到 acid 你可以 generate 一个 ATP， 对不对 ？generate 一个 ATP 咯。然后 succinate 到 fumarate， 我们刚才看到它会吐 FADH2 对不对 ？OK，fumarate 到 malate 会产什么东西吗？不会对不对？它只是一个 hydration。malate 到 OAA 会产一个 NADH，OK， 所以我 total 产了3个 NADH， 1个 FADH2 跟1个 ATP， 对不对 ？OK， 一个 NADH 可以给你几个 ATP？1.5 个。那 FADH2 可以给你几个 ATP？1.5 个，不合理嘛，对不对？所以 FADH2 可以给你1.5个 ATP，NADH 的话可以给你2.5个 ATP， 对不对？ 所以2.5乘3 7.5加1.5 9，然后再加产了一个 ，total 是10。OK， 所以一个 acetyl-CoA 它可以给你10个 ATP。Make sense？ 我刚刚这样应该大家听得懂吗？哈。Alright, so you get 10 ATP per acetyl-CoA. So you have 8 acetyl-CoA. So you have a total of 80 ATP. And then you have 7 FADH in the beta oxidation process. So 7 times 1.5, 10.5. 7 NADH, 7 times 2.5, 17.5. So total, okay, minus the 2 here, total is 106 ATP. One last thing I want you to think about, okay? Think about this, right? Think about this. Let's say, okay, let's say glucose is how many carbons? Glucose, 6 carbons, right? 6 carbons. 6 carbon fatty acid can give you how many ATP? You can count, okay? You can count. Is that going to be higher than glucose or lower than glucose? Higher, okay? 6 carbon fatty acid can give you more ATP than glucose, right? Why? Beta oxidation. What about, I'm looking for an answer where you compare glucose as a molecule and you compare fatty acid, 6个碳的 fatty acid as 一个 molecule. 这两个 molecule, 如果你比较这两个 molecule 的话，一看你就知道 fatty acid 可以给你比较多的 ATP， 为什么？这个是 6 carbon fatty acid， 对不对 ？Caproic acid。那 glucose 长什么样子？有人记得吗？如果我把它 linear 同样 linear 画出来的话 ，glucose 长什么样子？这边应该是一个 aldehyde。哎呦，这么厉害，还记得 ？OK， 剩下的呢 ？OH。OK， 所以如果我们这个是 glucose，actually 我不应该说 glucose， 但我 stereochemistry 没有画进去，所以我们就叫它 C6 的 sugar 好了。那你去看这两个分子啊，很明显的上面那个 molecule 能量一定会比较多，为什么？对，它比较 reduced。它的 state 比较 reduced， 它身上的那个 hydrogens 比较多，所以很多时候你在看这种 biochemistry 的这些 molecules 的时候，你光是看说这个 molecule 身上 oxygen 是多还是少，你大概就知道它这个东西的能量可以给你多少了。OK， 那 C6 acid 因为基本上它没有什么 oxygen，so 它的 energy 可以给你的会比较多。Compared to glucose, right? Glucose is pretty oxidized. Okay? Right, so be expected to be able to calculate how many ATPs that fatty acids can generate. Okay?